Fat digestion begins when chyme, an acidic fluid mixture of gastric juice and partly digested food, passes from the stomach into the initial section of the small intestine, the duodenum. Despite its name, the inner surface area of the small intestine is enormous. This is due partly to its great length, over 6 meters in humans, but principally to the numerous folds and tiny villi and microvilli that line its inner surface. Digestion requires many specialized enzymes as well as other secretions. The liver synthesizes bile, which aids in digesting lipids. The bile is stored in the gallbladder until it's needed to assist in fat digestion. When the chyme enters the duodenum, a hormonal signal causes the walls of the gallbladder to contract rhythmically, squeezing the bile into the common bile duct and down into the duodenum. The pancreas lies just beneath the stomach. It produces a host of digestive enzymes, including lipases, which are essential in fat digestion. The pancreas also secretes bicarbonate ions, which help to neutralize the pH of the chyme entering from the stomach. This is essential because intestinal enzymes, unlike stomach enzymes, function best at a neutral or slightly alkaline pH. Let's examine the process of fat digestion in greater detail. Fats are hydrophobic molecules, that is, they are not soluble in water. However, most enzymes, lipase included, are water-soluble and require an aqueous medium to function. Left to themselves, fat molecules would aggregate to form large globules that would present only a small surface area where the water-soluble lipases could act. Bile prevents this from happening by emulsifying the fat. The bile molecules have a hydrophobic end, which is absorbed into the fat droplet, and a hydrophilic end, which sticks out. This hydrophilic coating prevents the fat droplets from aggregating. These stabilized tiny droplets of fat are called micelles. Once stabilized in micelles, digestive enzymes from the pancreas can begin their work. Lipase breaks down the fats into fatty acids and monoglycerides. Because these products are lipid-soluble, they can easily pass through the plasma membrane in the intestinal mucosa and enter the epithelial cells. Once inside the cells, the fatty acids and monoglycerides enter the endoplasmic reticulum, where they're resynthesized into triglycerides, combined with cholesterol and phospholipids, and coated with a protein to form chylomicrons. The protein coat serves to make the chylomicrons water-soluble and to facilitate exocytosis. Chylomicrons serve as a shuttle to transport lipids through the body. The chylomicrons leave the mucosal cells via exocytosis and enter lymphatic vessels in the submucosa. From here, they're transported to the thoracic duct where they enter the bloodstream.